So as we all know, DNA is packaged into tightly packed, X-shaped chromosomes in eukaryotic cells. But how do we get into this tightly condensed conformation? Well, the DNA begins as a single strand, and then wraps around what we call histones, which are small molecules, onto which DNA wraps to become more tightly condensed. The first order of conformation is nucleosomes. So what are nucleosomes exactly? Nucleosomes are formed from four different histones: histone H1, H2A, H2B, H3, and H4. Now these nucleosomes are formed from multiple instances of these histones. So at first, these histones form dimers. Dimers are two of the same histone bonded together. So we have H2A, H2A, as well as H2B, H2B. Similarly, we have H3, H3, and H3, then H4, H4. So we have two different dimers here and two different dimers there. Now, these dimers will eventually bond together to form a tetramer. So here we have an H3H4 tetramer formed of one H3 dimer and one H4 dimer. And here we have an H2A H2B tetramer. Now, these two tetramers will then bond with one another to form an octamer. And octamers are the final histone shape that DNA wraps around. So ultimately, octamers are formed from one dimer of each histone, or two instances of every histone. Here we have DNA with an octamer, right about here. And then we see that the DNA wraps around the histone twice. And this would be about 146 nucleotide pairs around every octamer. Now, octamer, the octamer, plus the DNA which is wrapped around it, form what we call a nucleosome. And this is the first order of packaging. There are multiple histones along the strand, and the DNA wraps around it, just as we saw before. Now, one last thing I'd like to add here before maybe moving on to the second order of condensation is this chunk here between two histones. This chunk is called linker DNA. So it's just one term that I would like you to know because it is important to know. So the DNA between two histones, which is not wrapped onto any histone, is known as linker DNA. The second order of packaging is solenoids. Now, solenoids are formed from nucleosomes, as well as the last histone, which was not used in forming octamers, which is histone H1. So what happens is two nucleosomes with addition of H1 will turn onto themselves to form a solenoid. So if we draw a little figure here with a DNA strand right about here, a nucleosome here, an H1 histone here, we notice that another nucleosome gets tightly packed next to the original one. And the same goes here. So we have a bunch of nucleosomes right next to one another. This is just for reference, by the way. It goes on and on and on forever until the end of the chromosome. But this is what happens. So H1 makes two nucleosomes bind tightly to one another, forming solenoids, which are just tightly packed nucleosomes stacked on top of each other. The final order of packaging is scaffolding. So the now packed DNA will gather around scaffolding proteins, which will finally provide the X-shaped chromosome that we first saw in the beginning of the video. And that's it.